Right. First, I thank the organizers for the invitation. It's the last talk before some drinks, so bear with me the next 40 minutes or so. Today, I'm going to be telling you about some applications of single-cell multiomics and spatial transcriptomics to study plant-microbe interaction. So I prepared a bit of a general introduction. I'll do it quick, and I'm pretty much echoing what Emil had said at the beginning of this afternoon session. So as you all know, plants are essential for our lives. They provide what we eat. They also provide materials to build where we eat, because some of the previous speakers are telling where they're from. I'm from Japan. I don't have to emphasize how important photosynthesis is to our lives and to our planet. And at the Salk Institute, where I came from, there's an initiative called Harnessing Plants Initiative, where researchers are trying to engineer plants so that plants can absorb as much carbon dioxide as possible from the air and lock it in deep under into the root, deep under the soil to solve the climate change. So if this ambitious project is successful, our relationship with plants is going to be even stronger. With that said, an interesting fact to note here is that about 20% or even up to 50% of carbon fixed by photosynthesis is released into the soil. Why do plants do that? This is partially or maybe mainly because of the microbes in the soil. It's known that some of the carbon is released into the soil in the form of various metabolites, some of which are known to directly impact the soil microbiome. And again, 20% of photosynthesis is quite a bit of investment for plants, and this indicates how important interactions with microbes can be for plants' life. So what kind of microbes are there? There are pathogens that cause devastating diseases and huge economic losses. And that's why there have been a lot of research on plant immune system to learn how plants fight back against pathogens. And I think now it's a very exciting time because based on our understanding of the mechanisms of plant immune receptors, for example, we can now engineer plant receptors to make plants recognize virtually any pathogens as we wish. It's pretty much like vaccinating plants. And here, the molecular level understanding of immune responses or immune system is very important. Also, we heard a lot of symbiosis today. Nitrogen fixation is a very powerful symbiosis, but it's limited to certain plant species. So transferring this ability to other crop species is a holy grail in the modern or current plant biotechnology. And here again, basic understanding of the symbiosis mechanism is the key. And there are a bunch of others collectively called as commensals, simply because we don't know what they are doing. But there are a bunch of them in healthy looking plants in nature. And recently people started to show some beneficial functions of those commensal bacteria. So it will be interesting to learn the mechanisms of how they interact with plants. So my goal is to really understanding the fundamental principles underlying plant microbe interactions, whether they are pathogens, symbionts, or commensals, using and developing new technologies. And today I highlight single-cell multiomics and spatial transcriptomics, which is very good at resolving heterogeneity. And that's really useful to understand plant microbe interactions. Because plant microbe interaction is full of heterogeneity. Here's the model system that I present today. I use Arabidopsis leaf as a model host and Pseudomonas syringae as the model bacterial pathogen. Here's a confocal image showing this pathogen invading the host leaf. The pathogen is expressed in GFP, and you can see, the patchy distribution of bacterial colonies. Each colony would look like this, so packed with a bunch of bacterial cells. So, I think just this image will tell you a lot about how heterogeneous plant responses might be when, infected by pathogens. Here, I wanted to explain a little more about the various sources of heterogeneity. So first of all, plants are made of different cell types. Each has a distinct molecular system to respond against pathogens. And then cells can communicate with each other. Such cell-cell communications can be affected by spatial coordination between cells. And over time, each cell can change their responses. And also importantly, pathogens can change their distribution or activities, further affecting the cellular responses in a heterogeneous way, making things even more complicated. So to really understand the heterogeneity in plants at the molecular resolution, we decided to use single-cell omics, not only transcriptome analysis, but also epigenome to potentially understand the gene regulatory mechanism. And among many epigenome changes, we focused on chromatin accessibility. As you know, chromatin can be either closed or opened, and the open chromatin region can be bound by transcription factors and cofactors to regulate gene expression. So you could potentially learn about the gene regulatory mechanisms by looking at this modality. And as Mark mentioned, the chromatin accessibility can be analyzed at the single cell level using single cell ATAC-seq. For the transcriptome, of course, single cell RNA-seq can be used. And both approaches have been successfully applied to plant system, which is great. However, a challenge when you have those two separate data sets, the challenge is that linking those two modalities is quite difficult. Although there are some good fancy computational approaches to do this task, there's also one experimental approach to overcome this. And that is single cell multiomics, where you can analyze both modality, transcriptome and chromatin accessibility, from the same cell nucleus. So you don't have to worry about linking those two information from different separate cells. So we adopted 10x Genomics multiome assay. This is the first technology I introduced. So we sort of optimized nuclei isolation prep. So again, so Mark nicely explained, the difference between nuclei versus protoplast. But we decided to go for the nucleus approach. And using this system, here I highlight the sample, the conditions we use. You don't have to remember exact those strains, but we use 3 different strains. The idea is to maximize the situations that plants might experience. So this DC3000 is a wild type pathogen, which is virulent. It can cause diseases because this guy can suppress the immune system. The 2 other variants of DC3000 will trigger strong plant immunity called effector-triggered immunity. And this is why these pathogens don't cause diseases in plants. So plants win in this situation. We also have 4 different time points, relatively early stage of infection, again, to sort of diversify the situations plant cells might experience. Okay, so here's the UMAP summarizing all the cells profiled in this study, all those different conditions. And here the clustering was done based on only the transcriptome data. And based on the marker gene expression, we could identify major cell types like vasculature, mesophiles, epidermis, and so on. And by also looking at individual clusters for what kind of genes are expressed in each cluster, we could see some clusters are enriched with defense-related genes. So we could capture certain immune states, immune active cells and non-active cells and so on. Here I'm showing the actual gene expression, just one gene, ICS1, a very famous immune-related gene. This is a SA biosynthesis-related gene. And here you can see nice induction of ICS1 only in those 2 infection conditions where immunity is supposed to be strongly activated. And an interesting thing is that only a subset of those cells induced or expressed this gene strongly, which means immune activation is indeed heterogeneous upon pathogen infection. Here's the ATAC-seq data. So first, I just aggregated all the cells for each condition, mimicking bulk ATAC-seq just for the sanity check. And which is, so here, again, I'm showing ICS1 locus. And it's nice that we could see increase accessibility of the upstream region of this gene only in those conditions where the pathogen should have triggered strong immune activation. So this is consistent with previous bulk ataxic data. So the data quality looks good. And what's good about multiomics, single cell multiomics, is that now you can interrogate chromatin status in each cluster defined by the transcriptome data. And here you can see heterogeneous accessibility to this same region. And those clusters where you see increased accessibility happen to be those clusters where you saw the strong induction of mRNAs of ICS1. So there's a link between the accessibility to this accessible chromatin regions, ACR, and the nearby gene expression. So this kind of suggests certain regulatory mechanisms, right? So this region could be bound by transcription factors, which might drive this gene. So this is just one example, but we've scanned through whole genome, whole transcriptome to identify tens of thousands of such statistically significant links between ACRs and genes. Here I summarized all those thousands of genes that had at least one link to at least one ACR in at least one of those conditions. And first of all, you can see condition-dependent linkage. For example, these genes show the linkages only when infected by immune triggering bacteria. And when I look at those genes, these are mostly enriched with defense-related genes, which is good to see. One example is CBP60G. This is a famous transcription factor related to immunity. And this gene had multiple accessible chromatin regions linking or correlating with its expression. So it may be that multiple transcription factors are binding to those regions to give tight regulation on this important immunity. So the next question is, what kind of transcription factors are binding to those regions? So we did different kinds of motif enrichment analysis based on our existing motif database. Here, I just show you one example using WORKY46 as an example. This is, again, a defense-related transcription factor. So we calculated what's called motif activity score, which is calculated for each cell. And the cells with high motif activity score means that in those cells, WORKY46 binding sites are more open genome-wide, than just random chance. Whereas the cells with low score means that in those cells, WORKY46 binding sites are more close. So the WORKY46 might not be able to do its job. So in this case, those cells, we can hypothesize that maybe WORKY46 is doing its job in those cells. And we can actually look at the transcriptome data. This is epigenome data to ask whether WORKY46 itself is expressed in those cells, each cell. And of course, I brought the best example. So in this case, we saw a very nice overlap between mRNA. Sorry, something is wrong. So here, WORKY46 WORKY46 mRNA expression is shown here. So we can see the very nice overlap between mRNA expression of WORKY46 and the motif enrichment, motif activity of WORKY46, supporting that WORKY46 is indeed functioning in those cells. The next question is, what kind of genes are regulated by WORKY46 in individual cells? For this, we can use the linkage information. We can ask to which genes WORKY46 motifs are linked. And in this way, we predicted dozens of potential WORKY46 targets, which include a lot of known defense-related genes, which is nice. But also there are genes previously not characterized in the context of immunity. So they may be novel immune-related genes, which can be tested in the future by using mutant analysis. So this approach really identified the wealth of gene regulatory logic, including transcription factor, potential cis-regulatory elements, and target genes at the single cell resolution in this time course pathogen infection samples. So I only showed the WORKY46 as an example, but we've scanned through all the 465 motifs available today and identified a lot of potentially interesting gene regulatory modules, which is something I'm currently kind of validating now. Okay, so I'm switching gears to tell you about spatial analysis. So the spatial gene expression regulation has been known to be important, or at least involved in plant defense. So this previous study employed 2 transgenic reporter lines for 2 defense-related genes, PR1 and BSP2. Are we placed that? Okay. And they infected the plants. Oh, okay. They infected plants with the same pathogen as we used. So this is the infected region. As you can see different spatial and also temporal expression patterns between these 2 defense-related genes, which is not necessarily captured in this more static single-cell multi-omic data. No spatial information or temporal information. So, obviously the spatial gene expression analysis is important, but the limitation of this kind of approach is it's very low throughput. You could maybe chase 1 or a few genes at once, but not hundreds or thousands of. That's why we turn to spatial transcriptomics, specifically MERFISH. This is the technology number 2 in this talk. MERFISH is a massively multiplex single molecule imaging analysis that can capture hundreds of RNAs, hundreds of different genes at the single cell resolution on the tissue section. So we use the same kind of samples, the pathogen-infected Arabidopsis, at this nice longitudinal section, which is done by my postdoc colleague, Travis, and then applied MERFISH. So we use MERFISH instrument called MERScope, which currently allows us to target up to 500 genes. So I curated 500 genes, including leaf cell type markers, well-known immune-related genes or any other stress-related genes. And also to give some randomness, I also selected a lot of sort of random transcription factors, many of which haven't been analyzed or characterized. So here I'm showing some of the selected transcripts, maybe 10 or 20 transcripts. And we did the same kind of time course using this immune triggering pathogen and 4 time points, and this is control, no pathogen. And just looking at these images, same set of 20 or maybe 10 genes, you can see massive transcription reprogramming after pathogen infection, which is nice. And here I'm showing just 1 gene, LD1, a known immune-related gene, and you can see nice induction over time. And it's got a nice control that without pathogen attack, we didn't detect the transcript of LD1. So MERFISH is working pretty well, the low background signal. But the next step is to analyze all those 500 genes at once in a quantitative way at the single cell level. And this required a lot of optimization in the analysis pipeline. Oh, okay, something is wrong here, but let's see how it works. So one major challenge was cell segmentation. So based on the image data, we need to know where all the cells are. So the analysis was spearheaded by our rotation student, Alex Monell. What we did was we trained a deep learning model to learn or predict cell boundary structure purely based off the transcript distribution. So we didn't use any nuclei staining or cell boundary staining. We just used the transcript distribution to successfully predict the cell structure. And this allowed us to count each molecule, each dot in each cell to get this cell by G matrix, which is standard data type for single cell RNA-seq. In addition to this, we have this spatial matrix for each cell, X, Y, Z. In this case, Z being very small because it's a very, very thin tissue section. Okay. So this allowed us to do any, basic single cell RNA-seq analysis like dimension reduction, clustering, And what we did was we integrated all those merfish data with single cell multiome data that I showed earlier, using those shared 500 genes, okay, to get a unified map of all the cells profiled in these two assays, multiome and merfish. And you know, as you can see in these two color UMAP, these two assays kind of mix quite well. And then, okay, we did what's called label transfer, where we transferred this cluster label, cluster annotation, defined by the single cell multiome to individual merfish cells. And what's good about this is that now we can use merfish cells as a spatial anchor to spatially map all the multiome cells, which is whole transcriptome, not only 500 genes, it's 20,000 genes. And also it has chromatin accessibility information such as motif activity. So this sounds good. So we wanted to test whether it works at all. So we first tried to spatially map predicted major cell types predicted from the single cell multiome data, no spatial information. Based on the marker gene expression, we predicted 3 major cell types: epidermis, mesophiles, and vasculature. So we integrated merfish data with this data with merfish and did spatial mapping of these cells. And I hope you can see that it worked pretty well. So if you look at the yellow cells, it shows very nice vasculature structure. And most of the cells are mesophils, as we section through the middle of the leaf. But there are some epidermal cells on the periphery of this tissue section, because the tissue sections are not completely flat. So we have some epidermal sort of contamination. But this is nice to see, that the data makes sense. So we are convinced by this, or motivated by this result, and try to map any other information such as transcriptome and epigenome. If you remember that I showed that we predicted many potentially new gene regulatory mechanisms, WORKY46 as an example. So here we spatially mapped WORKY46 motif activity, which is epigenome data. This is the immune activated tissue, and you see heterogeneous distribution of cells with high WORKY46 motif activity. Here we show the expression of one of the predicted WORKY46 targets, ALD1, which is here. This is the mRNA expression, and it also showed kind of heterogeneous expression pattern. It might not be easy to see, but they actually overlap quite well. If you look at maybe somewhere here, high motif activity and high ALD1 expression. So we could potentially spatially map all those predicted gene regulatory mechanisms to, which enables us to study novel gene regulatory mechanisms at really high resolution. So lastly, we also wanted to understand the temporal dynamics of immune responses. And for this, we applied pseudo-time analysis, where we infer the trajectory of gene expression changes. So we apply pseudo-time analysis only to the mesophil cells, this is a pseudo-time prediction. So those cells are shows low value, high value here. Our interpretation is that the cells with low pseudo-time values are the cells that are not immune activated or immune activated not so long ago, while the ones with high pseudo-time values are the cells that are immune activated quite long ago. Is that accurate? We can actually ask this because we have the real-time course data. Here's the real-time course data. Now you can see more blue cells at early time point for no bacteria, no pathogen infection, but more red cells at later time point, sort of supporting this interpretation of the data. So now, having modeled the temporal dynamics, we can spatially map this information based on the integration with merfish data. This is pretty exciting because now here you can see the emergence of those light color regions over time. And these are immune active regions. And they grow over time. And then at the latest time point, I believe multiple immune active regions are kind of combined and collapsed and further changing their transcriptome responses. So we could successfully model the spatial temporal dynamics of plant immune responses at the single cell resolution. And I hope this molecularly defines map of immune responses will allow us to mechanistically dissect plant immune responses against pathogens. And we want to analyze, for example, how individual cells respond to this pathogen and how they communicate with each other to spread immune responses to stop pathogen growth. Okay, so I hope I could show that spatial transcriptomics is exciting. However, no technology is perfect, as you must know quite well. One big limitation with the current, at least commercially available spatial transcriptomics, is that you have to cut sections, and tissue section is hard. First of all, it's not easy, especially when your tissue is very tiny, like Arabidopsis leaves. Even if you can cut the perfect section, you're going to lose all the rest of the tissue, which may contain important cell types, or in my case, some pathogen cells. So if you lose this information, the data interpretation on this thin tissue section is going to be really tricky. So that's why we need some sort of spatial transcriptomics kind of assay, which can work in much, much thicker tissue, ideally whole mount tissue. So that was the motivation behind the development of Phyto-map, which is a multiplex and single cell 3D spatial gene expression analysis in whole mount plant tissue. I think I have some time to quickly explain how it works. So Phyto-map, so first you fix the whole mount tissue, so all the mRNAs don't move from their original positions. And the key step in Phyto-map is that all mRNAs are replaced with DNA probes. So you hybridize DNA probes, which has barcode sequence specific to each gene. And then this barcode containing DNA probes are amplified for hundreds of times in situ, and then again fixed tightly. So after this, those mRNAs are completely replaced by amplified DNA probes at their original positions. And then we perform single molecule FISH targeting gene-specific barcode sequence. And because the barcode sequence has been amplified for hundreds of times, it can incorporate a lot of fluorescent probes. And so this will glow really bright. This is important for deep tissue imaging in such a potentially autofluorescent plant tissue. And in each imaging round, you can detect 4 different target genes, which is shown here. Each spot is derived from single molecule. Different color means different genes. And the good thing is you can use exactly the same tissue and remove the fluorescent probes, and then rehybridize you can redo FISH again, targeting another set of 4 genes, which is here, and then you repeat again and again until you have painted all the target genes you wanted to visualize. So here I'm showing, okay, only one of them is moving, but okay, no. All right, so these are Arabidopsis root tips. Again, it's a tiny tissue, so it's very difficult to cut nice section. So here we targeted some of the known or predicted cell type marker genes or tissue region marker genes. So you can kind of see specific expression patterns already here in these 3D images. Some blown up images to show the specificity of Phyto-map with cell wall staining. So here, thank you. So here you can see specific expression of different colors. They are predicted cell type markers from a specific layer of the tissue. So the Phyto-map is pretty target specific. Okay. And so I have more information in this recently published paper, but here I just briefly summarize some key features of Phyto-map. So I believe this is currently the only approach which can visualize dozens of genes at once in whole mount plant tissue. And the protocols are pretty straightforward. It's similar to single molecule FISH. So it takes 4 to 5 days for sample prep. And if you have confocal microscope, you can do imaging. And the good thing is the cost is quite inexpensive compared to other spatial transcriptomics platform. Although these technologies do different things, in my estimate, it costs only $80 per experiment, which can accommodate more than 5 root tip. So you can scan through maybe 5 different genotypes if you want. And another important feature is that it's transgene free. You don't have to make transformation, the transgenic lines, which takes time. But also, this means that Phyto-map could be applied to any plant species, including model plant species where you can't transform regularly. So I hope many people would use this technique in their favorite model system. Okay, so in the future, in my new lab, I'd like to combine those techniques I introduced today. For example, a single cell multiomics, which will allow us to characterize cell populations and reveal key marker genes, potential marker genes, which can then be visualized using Phyto-map in their native context, the 3D tissue context. But also, we'd like to further advance Phyto-map by adding more features, such as multimodal assays, which means not only visualizing mRNAs, but at the same time, visualizing proteins or metabolites or even microbes to get as much information as possible. I think there will be a lot of interesting discussion, about how we could integrate Phyto-map with technologies available here, like spatial metabolomics. Some of the key or high priority question in the next few years will be, how do plant cells initially infected by pathogens respond? This is a very simple question, but we still don't know. Probably the next question is, how do plant immune responses spread from these initially infected cells to the neighboring cells? And after understanding all these cell populations, what their functions are, how they are regulated, such knowledge will allow us to engineer plant tissue in a very precise way, engineering a specific cell population in a specific way, which I think can open the door for precision breeding, which I think some people have discussed during this meeting. Okay, so this is my last slide. So towards this goal, just here, I wanted to introduce another resource we've developed in the ECHO lab at Salk. So we created single cell transcriptome atlas of this model plant Arabidopsis that covers the entire life cycle of this plant, which means we started from the seeds, the germinating seeds and seedlings, rosettes, different stages, stems, flowers, and sleeks, and seeds inside the sleek. So we call it a seed to seed atlas. And we identified hundreds of transcriptionally distinct clusters. Some of them are known cell types, but many of them are just unknown cell states. So today, I mostly focused on matured leaves infected by pathogens, but plants have way more cell types and states from which we can learn about new molecular mechanisms, which could then be exploited for the engineering of plants in the future. Okay, so with this, I want to thank all the people involved in the studies that I showed today, especially my mentor, Joe, and also collaborators. And lastly, a shameless plug from my side that I'm now recruiting some postdocs and also students and technicians in any positions will be available in the future in my upcoming lab at the Century Laboratory in the UK. So if you know somebody who may be interested, it would be great if you could help spread the word. Thank you so much.
Thanks for the fantastic talk. Questions?
A great talk, really, really interesting. Two questions. The first is a technical question. Your experiments that you did with Pseudomonas syringae, the images suggested to me that you used a syringe infiltration to inoculate, which of course is not the way that the bacteria normally infects in the wild. It infects through stomates. So I wanted to clarify that. And have you gone back and look at stomatal infection, because I think it'll be more relevant to what actually happens in the real world. And then the second question is just how expensive was this multiomics experiment that you did with Pseudomonas? I think that it's great technology, but I think for the common man that's not supported by Howard Hughes, it's a limiting factor. So.
Thank you for the question. So the first question is the infection strategy. You're correct. So we syringe infiltrated pathogens. This is simple. I initially tried to do spray inoculation, which, as you know, triggers more subtle responses. And at that time, like 3 years ago, we couldn't capture very robust immune responses with this approach, probably because very few cells are responding immune responses. And again, it's very costly to sequence a lot of cells. So that's one of the reasons. Although we did syringe infiltration, we infiltrated bacteria at very, very low dose. So I expect only, it's basically fewer bacterial cells than plant cells. So only some of the plant cells should be in direct contact with pathogen. That's how we try to create the heterogeneity. And to the second question, yes, it's very expensive. I don't know if I should give a specific number, but it could be $10,000 per sample, including sequencing. So it's pretty expensive. But there are new technologies that makes those expensive assets more inexpensive, such as recently Bob Schmitz posted a preprint called Sci-Fi Ataxic, where you can pre-index the cells so that you can kind of hyper-rolled. You can put a lot of cells to the 10x genomics platform, like a 10 times or maybe 20 times more, which can lower the cost by significantly. So I hope those tricks hopefully will be applied to many other systems to sort of make those single cell technologies more accessible. In addition, if somebody has done those expensive single cell data, one could use Phyto-map, which is much more inexpensive approach to confirm or their spatial gene expression patterns for the selected genes based on the single cell data, because you don't really need to visualize 20,000 genes. You can kind of select the most interesting 50 genes, for example. Then you can you need only $80 to see that.
Hello. Hi. Great talk. Thank you. I have a question about the Phyto-map. So I think it's a great idea to do like whole mount spatial mapping, but since you're using fluorescent labeling, like the signals are essentially fluorescence, so it's still going to be limited by the the thickness. So you're going to use laser to penetrate the sample in order to have the fluorescence emission. So it's going to be limited by the thickness of the tissue. So you actually can't go very thick in that sense, right? So like what, and then there are like as the deeper the tissue goes, the power of the laser will also decrease. So I'm just wondering, like, what are some, future, I guess, like options that you're exploring to kind of overcome these, I guess, shortcomings?
Thank you for the question. Yeah, as you said, Phyto-map is not free from, the general limitation in imaging. But you can, it's a fixed tissue, so you don't have to worry about killing cells by, turning up the laser. So you can do really aggressive imaging. You can also use tissue clearing. We used Clear-C, which makes the tissue transparent, which obviously helps. And also, there are many other techniques where you can basically digest all the cell walls, for example, to remove potential autofluorescence. And if you mount the tissue into the gel, the transparent gel, this allows you to go really deep. One last thing is you could also use light sheet, which, there are good light sheet which can scan the whole mouse brain, for example, or whole mouse body, which is really big tissue. So you could apply such technology, cutting edge microscopy to see the big tissue with Phyto-map.
Hey, great talk. I am just curious about one thing regarding Phyto-map, because you guys are identifying transcripts using probes, right? I was just curious to know what portion or which portion of the transcript you are mapping or targeting using the probe, because that could lead to false positives, even if the probe, a smaller probe can attach to a smaller or spliced transcript or something like that.
Yeah, that's a great question. So I think I will first tell you about the false negatives, because we target multiple regions in the transcript. I use 4 different regions to maximize the capture rate, because there should be some dropout if you just only target one. So this can reduce the false negative. For the false positive issues, we use a pair of 20 base pair probes. So in total 40 base pairs. So the Phyto-map needs both probes to be landing on the same region. So this gives you certain specificity. But again, it's a hybridization-based approach, so you need to really optimize the condition to optimize the sensitivity versus, let's say, how it's called, accuracy.
Makes sense. Thank you.